Hello, this lecture will be about agglutination. Agglutination in Latin, agglutinare, means to glue to. So agglutination methods involve the clumping of small particles aided by agglutinins. Agglutination is the visible aggregation of particles caused by combination with specific antibody. And agglutination differs from precipitation in that you have a soluble antibody and an insoluble or particulate antigen, or an insoluble antibody and a soluble antigen. So one part will be soluble and one part insoluble, either the antigen or the antibody. Soluble particles are made insoluble when they combine with latex particles or red blood cells, etc., like dyes or liposomes. Red blood cells and bacterial cells have a slight negative charge that will repel each other, and we'll talk more about that a little later in this lecture. Agglutination, just like precipitation, is a two-step process that involves sensitization, or the initial binding, followed by lattice formation, or the formation of large aggregates that we can see. The first reaction of agglutination involves antigen-antibody combination through single antigenic determinants on the particle surface, and this is called the sensitization step. The first step is very rapid and reversible. And here is a picture representing sensitization. So you can see that the antibodies are attaching to the corresponding epitope on, for example, this is a red blood cell membrane. There's no visible clumping in this phase, and the red cells must be close enough for the FAB portion of antibodies to bind and make bridges between the cells. The second step is formation of cross-links, and that's when they form visible aggregates. At this point, we can begin to see some clumping. The antigen-antibody complex has a stable bond, and each stage of this reaction can be manipulated to test for either antigen or antibody in the clinical lab, and so we use this method of testing quite a bit. Lattice formation, or the second stage, is the sum of interactions by antigen and antibody, and depends on the concentration of antigen and antibody, and the physiochemical factors, such as the ionic strength, the pH, the temperature, things of that nature. Red blood cells and bacterial cells have a slight negative charge and will repel each other. Here's a picture of lattice formation, and this represents what occurs during the second stage of agglutination. Antibody molecules cross-link with red blood cells, forming a lattice that results in visible clumping or agglutination. So this illustration of stage 1 and 2, these pictures that I put in for you, it's actually heme agglutination because red blood cells are used as the insoluble particle, but the sensitization and lattice formation phases work the same, and you can use different carrier particles, and we're going to learn about those. In agglutination, sensitization can be affected by the antigen-bearing surface and the affinity and avidity of an individual antibody. That determines how much antibody remains attached. Now remember, back to the antibody structure and function chapter, what does a valence of 10 mean? That means that an antibody has 10 binding sites. And we remember that IgM has a potential valence of 10. So it is over 700 times more efficient in agglutination reactions than IgG that only has a valence of 2. And here we have an example of the IgG with a valence of 2 and the IgM with a valence of 10. Antibodies that produce such reactions are often called agglutinins, and types of particles that can be used in these reactions include erythrocytes, bacterial cells, and inert carriers such as latex particles. And these latex particles that can be used are like microscopic all the way up to the size of an individual Mardi Gras bead. Each particle must have multiple antigenic determinant sites, which are cross-linked to sites on other particles through the formation of antibody bridges or lattices. So all of this depends on the ability to have multiple sites on the carrier particle that can be cross-linked with antibodies to form lattices and aggregates that are big enough for us to see and measure in the clinical lab. Agglutination reactions have a high degree of sensitivity, and there are different classifications of categories of agglutination reactions, and we're going to talk about each one of these: direct, passive, reverse passive, agglutination inhibition, and co-agglutination. The antibody must be able to bridge the gap between the cells so that one molecule can bind to a site on each of two different cells. So this figure 9.1, it's in your book, and it depicts this two-stage process. So there's antibody that's floating around free, then we have an antigen with multiple determinants, and then you get this agglutination reaction. And here's a picture of the red blood cell and the net negative charge that it has around it. So it's called the zeta potential. And don't memorize this. I ripped this off the internet. This is not figure 5 from your book. But this red blood cell, it has this net negative charge, so it repels other red blood cells. So this is one thing that helps your blood not stick together while it's in your body. The surface charge must be controlled for lattice formation or visible agglutination reaction to take place, right? Because if we're trying to see if an antibody or antigen is present, we don't want our red blood cells to repel each other if one of those is present. So how can we do this? One means of accomplishing this is by decreasing the buffer's ionic strength through the use of low ionic strength saline. We call this LIS, and this is a very common reagent used in blood banking. So we just said that red blood cells have a slight negative charge. Well, LIS and another reagent called albumin act as a potentiator, and this acts as a potentiator because it can reduce the charge around suspended red blood cells. So the repulsive negative charges are dispersed, which increases sensitization and agglutination. PEG, or polyethylene glycol, also increases the viscosity of solutions, thereby enhancing the agglutination reactions. And so these are all reagents in a blood banker's toolbox that they can use to enhance and encourage agglutination reactions. Because if there is an unknown antibody in our patient sample for blood bank, we want to do everything we can to get that to show up in the laboratory. So that's why we use these things. The addition of albumin in a concentration of 5 to 30% helps neutralize the surface charge and allows red cells to approach each other more closely. So this is a 7.5 or 6.5% protein suspension. So albumin, remember, is the biggest protein in serum, and that's across, it's very common in a lot of animals. So this is just a protein suspension that helps reduce that negative charge. Other techniques that enhance agglutination include increasing the viscosity by adding PEG, using enzymes, and if you remember back to the antibody structure and function lecture, we talked about papain and pepsin that cleave off certain parts of the antibodies to allow reactions to take place. Agitating, so shaking the tubes just a little bit, or centrifuging to spin everything down to the bottom of the tube to encourage agglutination. Also altering the temperature or the pH, because we're going to learn that some antibodies prefer to react at body temperature at 37 degrees Celsius or 98.6 degrees Fahrenheit, and some prefer cool temperatures, either room temp or even 4 degrees Celsius. Agitation and centrifugation provide a physical means to increase cell-to-cell contact to increase agglutination. So that's actually like shaking the tubes just a little bit and then centrifuging it to pull everything to the bottom of the tube. The temperature must be considered because IgG antibodies agglutinate best between 30 degrees to 37 degrees Celsius, and IgM antibodies agglutinate best between 4 degrees to 24 degrees Celsius. Now, we just said that we know body temperature is 37 degrees Celsius, so 30 to 37, that's pretty warm. But 4 degrees to 24 degrees Celsius, 4 degrees Celsius should be the temperature of your refrigerator at home. So those give you a point of reference. Optimum pH for most reactions is between 6.7 to 7.2, and of course, there are exceptions. Agglutination reactions can be classified into several distinct categories: direct, passive, reverse passive, agglutination inhibition, and co-agglutination. Most agglutination tests are qualitative. That means they just detect positive or negative, presence or absence of the antigen or antibody for which you are testing. Dilutions can be used for semi-quantitation. So a titer is the last dilution still exhibiting a visible reaction. Direct agglutination occurs when antigens are found naturally on a particle. One of the best examples of direct agglutination involves known bacterial antigens used to test for the presence of unknown antibodies in the patient. One example is Salmonella. The antigens used in this procedure include Salmonella O, or somatic antigen that's part of the Salmonella cell wall, and the H flagellar antigen. So that H antigen is part of the flagella on Salmonella. And this is exactly what it looks like. So for an example of a direct agglutination test, and you can do so many different kinds of bacterial typing. I mean, just if you can think of an antibody for it, or you can find an antigen for it, you can use the opposite to test for it in a patient's serum. So the principle of direct agglutination is that you combine an insoluble particulate antigen with its soluble antigen. So what we have here is we have bacterial particles with the antigen, and they've been combined with patient serum that has the antibody. So the antibody attaches to these carrier particles with the antigens, and they clump together, and so you can see it. So here's a positive reaction. This is what we can actually see, these clumps. And over here is a negative reaction where everything stays like a uniform white, no clumps. So other types, you can use Vibrio cholera, Salmonella, group strep testing like group A, group B. When you do a culture, you do direct agglutination. So we use this type test method all the time. If an agglutination reaction involves red blood cells, then it is called hemagglutination. And the best example and most common of this occurs in the ABO blood group typing of human red blood cells. Positive reactions can be graded to indicate the strength of the reaction. So if you add your patient red cells to the antiserum, you centrifuge it, and if you have if you shake it, this would be agitation. If you shake it and have one solid clump, this would be a 4+ reaction. If you have several large clumps, this would be a 3+ reaction. If you have several smaller clumps, that's a 2+. And if you have barely discernible clumps, that's a 1+. And negative smooth suspension is negative. So let's look at our blood groups over here. So this clumping right here under the A, this would be a 3+ reaction. This B would be a negative reaction. AB, this one right here, this would be a 3+. So there are no 4+ reactions here, but this is probably a 3+. These are probably 2 or 3. You just learn how to grade reactions when you go to clinicals, and you learn how to do it like they do. We have a saying that when you get to clinicals, when in Rome, you do what the Romans do, as long as it's not really wrong. All right. This type of hemagglutination reaction is simple to perform, it's relatively sensitive and very easy to read. A titer that yields a semi-quantitative result can be performed in test tubes or microtiter plates by making serial dilutions of the antibody. So let's take a look at this example here. You have a 1 to 20 dilution, a 1 to 40 dilution, a 1 to 80 dilution, a 1 to 160 dilution, and then 1 to 320 and 1 to 640, and the control is clear. So there's no reaction in the 1 to 320 and the 1 to 640. So the last tube with any visible agglutination is 1 to 160, so your titer would be 160. Agglutination reactions can also be passive or indirect, and this method uses particles that are coated with antigens not normally found on their surfaces. Again, particles used can be red blood cells, latex, gelatin, silicone parts. They can all be used for this purpose. The use of synthetic beads or particles provides the advantage of consistency, uniformity, and stability. So what this does is this takes any variation out of the soluble particle, and it makes it very uniform, consistent, and stable. So it's very, very precise and more accurate. Reactions are easy to read visually and give quick results in minutes. Latex particles, they are better than red blood cells. They are inexpensive. They're relatively stable. They are not subject to cross-reactivity with other antibodies, and a large number of antibody molecules can be bound to the surface of latex particles. So the number of antigen binding sites can be very large. In addition, the large particle size makes reading of the test very easy. Passive agglutination tests have been used for things like rheumatoid factor, anti-nuclear antibodies or lupus, group A strep, and a whole bunch of different viruses. So passive agglutination tests start with a carrier molecule like a latex bead and a soluble antigen, and those get stuck together, and then you add an antiserum, which has an antibody to these antigens. So the carrier molecule absorbs the antigens, and then the antibodies stick to the antigens on the carrier molecules, and that's what causes the lattice. So it's indirect. The antibodies are to these antigens that are stuck on these carrier molecules. So this is called passive agglutination. In reverse passive agglutination, antibody instead of antigen is attached to the carrier particle. The antibody must still be reactive and joined so that the active sites face outward. So the FAB portions have to be outward, ready to bind with antigens. This type of testing is often used to detect bacterial antigens such as group B strep, Staph aureus, Neisseria meningitidis, group A strep, Haemophilus influenza, rotavirus, Cryptococcus, Mycoplasma pneumoniae, or Candida albicans. So the tests that they do for newborn meningitis, it's a panel, and it has little bitty tiny latex beads, and it's looking for this stuff all at the same time. Rapid agglutination tests have the widest application in urine, spinal fluid, and serum. And this figure on the next page shows the differences between passive and reverse passive, but I wanted you to just concentrate on reverse passive agglutination. Remember, so in reverse passive, you have antibody already stuck on your carrier molecule. Then you add soluble antigen, and this is how the lattice forms. It's really a cool method. The use of monoclonal antibodies has greatly cut down on cross-reactivity, but there's still the possibility of interference or non-specific agglutination or a false positive. So how are monoclonal antibodies made? Think back to your previous lectures. You have a plasma cell that makes antibodies plus a cancer cell, and you squish those together and it makes a hybridoma, so a cell that makes one type of antibody for a long time. Such tests are most often used for organisms that are difficult to grow in the lab or for instances when rapid identification allows treatment to be initiated very quickly. In all of these agglutination reactions, rheumatoid factor will cause a false positive, as it reacts with any IgG antibody. So we're going to talk about rheumatoid arthritis in our autoimmune chapter, but rheumatoid factor is it's an antibody to the Fc portion of IgG molecules. So when somebody has rheumatoid arthritis, their body is making antibodies that attack their own IgG antibodies, especially that Fc portion. And those antibodies precipitate out in their joints, and that's what causes the inflammation and swelling. So you have all this extra antibody, and a lot of the autoimmune tests, autoimmune diseases will interfere with the agglutination test, but specifically this rheumatoid factor is rough on agglutination tests. Agglutination inhibition is just what it sounds like. With agglutination inhibition reactions, a lack of agglutination is a positive reaction. So let's go through this pretty slowly. Agglutination inhibition reactions are based on competition between particulate and soluble antigens for a limited number of antibody combining sites, and a lack of agglutination is an indicator of a positive reaction. So in these tests, agglutination is a negative reaction. So why? Let's think through this. Because this type of test or reaction involves haptens that are complex to proteins. The hapten protein conjugate is then complex to proteins, is then attached to a carrier particle. So the hapten protein conjugate is attached to a carrier particle. The patient sample is first reacted with a limited amount of reagent antibody that is specific for the hapten being tested. Indicator particles that contain the same hapten one wishes to measure in the patient are then added. If the patient sample has no free hapten, the reagent antibody is able to combine with the carrier particles and produce a visible agglutination. In this case, however, agglutination is a negative reaction, indicating that the patient did not have sufficient hapten to inhibit the secondary reaction. Hemagglutination inhibition reactions use the same principle, except red blood cells are the indicator particles. Very interesting. So I've found this picture to illustrate hemagglutination inhibition. Okay, so if you're looking for a virus or antibodies to a virus, you would have the viral particles, then you would add your red blood cells, and then those viruses would attach to the red blood cells, forming a lattice, and then you would just get this no homolysis, right? So then you have a hemagglutinating virus, then you add the antibody to the virus, then you have antibody antigen complexes, you add the red blood cells, and then you have no binding, so there is no lysis. So you have hemagglutination inhibition. This type of testing has been used to detect antibodies to certain viruses such as rubella, measles, mumps, flu, parainfluenza, hepatitis B, herpes, respiratory syncytial virus, and adenovirus. Coagglutination is the name given to systems using bacteria as the inert particles to which antibody is attached. Staph aureus is most frequently used because it has a protein on its outer surface called protein A, which normally adsorbs the Fc portion of antibody molecules. So the Fc portion of antibody molecules can stick on this protein A on the surface of Staph aureus. The active sites, or FAB, faces outward, and they're capable of reacting with specific antigens. These particles exhibit greater stability than latex particles and are more refractory to changes in ionic strength. However, because bacteria are not a color, reactions are often difficult to read. So here's a picture of our coagglutination picture. We have our Staphylococcus A bacteria with the protein A on the surface. So you have specific antibody that then attaches, so the Fc portions attach to the protein A. Then you add red blood cells that may or may not have the antigen, and then you get this big particulate agglutination. The anti-human globulin test, also known as the Coombs test, is a technique that detects non-agglutinating antibody by means of coupling with a second antibody. So what we're looking for is we're looking for an antibody to our antibody. That was what we use to look for unexpected antibodies. There are two reagents that can be used for the Coombs test. One we call the AHG for IgG. So the AHG, the anti-human globulin, that just means you're using IgG alone, the anti-IgG. Or poly, which is a solution that contains an antibody to IgG and an antibody to complement components. So this is what happens in the direct Coombs test. In the direct Coombs test, you are looking for antibody coating the red blood cells in vivo. So you have red blood cells that's coated with antibody. Then you add your anti-human IgG Coombs reagent, so it's an antibody to human antibody. And then if the red blood cells are coated with antibody, you get this big agglutination sample. So this is the direct antiglobulin test used to demonstrate in vivo attachment of antibody or complement to an individual red blood cells. The key component of the test is antibody to human globulin that is made in animals or by means of hybridoma techniques. Such antibody will react with the Fc portion of the human antibody attached to red blood cells. Agglutination takes place because the anti-human globulin is able to bridge the distance between cells that IgG alone cannot. Strength of the reaction is proportional to the amount of antibody coating red blood cells. The direct antiglobulin test, or DAT, is used to demonstrate in vivo attachment of antibody or complement to an individual's red blood cells. This test serves as an indicator of autoimmune hemolytic anemia, hemolytic disease of the newborn, sensitization of red blood cells caused by the presence of certain drugs, or a transfusion reaction. So these 4 examples right here are all examples of diseases when your red blood cells become coated with unknown or unexpected antibody in your body. Hemolytic disease of the newborn is when the newborn's red blood cells are being attacked by antibodies from the mother. The attack begins while the baby is still in the womb, and it's caused by an incompatibility between the mother and baby's blood. The DAT is called direct because the red blood cells are tested directly as they come from the body. And this DAT and AHG stuff is very important. The indirect antiglobulin test, or IAT, or indirect Coombs test, is used to determine the presence of a particular antibody in a patient. Or it can be used to type patient red blood cells for specific blood group antigens. Washed red blood cells and antibody are allowed to combine at 37 degrees Celsius, and the cells are then carefully washed again to remove any unbound antibody. When anti-human globulin is added, a visible reaction occurs where antibody has been specifically bound. This test is the most often used to check for the presence of clinically significant alloantibody in patient serum when performing compatibility testing for a blood transfusion. And right now, you probably know that there is the A blood group, there's the B blood group, there's the O, there's the AB, and there's also the RH. Well, in blood bank, you're going to learn about so many other blood groups and antigens. You just have no idea. It's so exciting. But we have to check for these unexpected alloantibodies when we are doing compatibility testing for blood transfusions. So here's an example or an illustration of the DAT and indirect Coombs. So here's the indirect Coombs. That's a better picture. So for the indirect Coombs, you are looking for antibodies in the patient sample. So patient sample, antibodies. So for this test, we have known types of red blood cells in bottles that we can use, and we mix the red blood cells with the patient's serum, our known red blood cells with the patient's serum or screening cells, and we look for agglutination. Sources of error are failure to wash the cells, because you have to wash between each step to remove any unbound antigen or any unbound antibody from your reagent tubes that you've added. Improper centrifugation, if you don't centrifuge enough or if you centrifuge too long at too high of a speed, that can cause a false positive. Or if you use expired or improperly stored reagents. And here's a picture of the direct Coombs versus the indirect Coombs. So for the direct Coombs, we're looking for antibodies coating the red blood cells in vivo. And for the indirect, we're looking for antibodies in the serum. Quality control and quality assurance are important, very important in the blood bank. Although agglutination reactions are simple to perform, interpretation must be very, very carefully done. Techniques must be standardized as to concentration of antigen, incubation time, temperature, diluent, the method of reading, and your sample solution rack or tray, your reagent rack, should be set up the same for everybody in the lab every day, all the time. The possibility of cross-reactivity and interfering antibody should always be considered. Cross-reactivity is caused by the presence of antigenic determinants that resemble one another so closely that antibody formed against one will react with the other. Most cross-reactivity can be avoided through the use of monoclonal antibody directed against an epitope that is unique to a particular antigen. And it's also, you should note that heterophile antibody and rheumatoid factor are two interfering antibodies that may produce a false positive result. So the heterophile antibody is something that you can get when you have Epstein-Barr virus, which causes mononucleosis. And that brings us to a conclusion of agglutination reactions.